Let's look at some ways we can distinguish between the different types of tissues that we find in animals. Animals have four major types of tissues. Epithelial tissues are found as the covering of other tissues. They're found lining things like the digestive and the respiratory tract. They provide a barrier and a layer of protection. In muscle tissues, the cells are able to actively shorten. Nervous tissue is used for sending signals, and connective tissue includes almost everything else. It includes some obvious things, such as tendons that connect one structure in the body to another, but it also includes things like our bones, our blood, things that you might not think of as a connective tissue. So if it doesn't cover something, if it doesn't get shorter, if it doesn't send a signal, it's going to be classified as a connective tissue. The best way to recognize epithelial tissue is to look for an opening. Because it covers or lines, you're going to have an open space either in the interior if it's lining a tube, or a big open space at the edge. So epithelial tissue is going to cover or line. We classify epithelium by the shape of the cells and the number of layers of cells. Simple epithelium just has a single layer of cells with one nucleus. This is the underlying connective tissue. You know it's epithelium because there's an open space, and then there's just one cell with one nucleus between that open space and the underlying connective tissue. A stratified epithelium has lots and lots of layers of cells and lots of nuclei between the underlying connective tissue and the opening. Stratified epithelium makes great protection. Pseudostratified is exactly what you might expect. It looks like it's stratified. There's lots of different layers of nuclei, but if you could follow the cells, there would be some short cells, there would be some long cells, and they all come down and attach to the same point. So there's really just one layer of cells, but the different heights and arrangements make it look like there's several. The other way we classify epithelium is by the shape of the cells. Squamous epithelium has squashed flat-looking cells with squashed flat-looking nuclei. Cuboidal epithelium is box-shaped, cube-shaped, and the cells tend to have nice round nuclei. Columnar epithelium has column-shaped cells. The nucleus within the cell is usually a long oval. Transitional epithelium is a little more complicated because the shape can change. It's epithelium because there's an opening. The cells here are nice and round, but transitional epithelium is found in places like the urinary bladder. As the bladder fills with urine, this whole epithelium can stretch out, and the shape of the cells will change and become flatter. To identify epithelial cells, we just put the words for the shapes together with the words for the number of layers. Simple squamous epithelium is used to quickly move substances across the cell and get them in or out of an area of the body. This is a simple squamous epithelium there, and you can see how flat and thin and squashed looking the cells around the opening are. This is also epithelium because there's an opening, but these larger cells are cuboidal cells. Stratified squamous epithelium provides lots of layers for protection. They're squashed flat cells. The cells at the top are usually dead and expendable, and they can fall off and be replaced by the division of younger cells underneath them. Cuboidal epithelium would be a single layer of roughly square-shaped cells with nice round nuclei. These look fuzzy because they have cilia on their surface. Those cilia would be used to move substances through that opening. Simple columnar would be a single layer of columns, and again we can see a layer of cilia up here on these column-shaped cells. This lining this opening. This is another layer of epithelium, single layer lining this opening. Pseudostratified columnar epithelium looks like it has lots of layers of columns, but there is really just a single layer. These also have cilia, so if you want a phrase to throw into a conversation, you can call it pseudostratified ciliated columnar epithelium. There are 3 different types of muscle tissue. Skeletal muscle is what we usually think of as muscle. It's what we use to move the body. Skeletal muscle has very long cells. The cells are so long that they're called fibers. The cells often have more than one nucleus, which ends up squashed to one side. And within the cells, we see these stripes or striations. These are caused by the regular arrangement of actin and myosin, the proteins that the muscle uses to contract and shorten. Smooth muscle has actin and myosin, but they're not arranged like they are in skeletal muscle, so we don't see the striation. The best way to recognize smooth muscle is that its fibers, its long cells, end in little points. So look for the little points and you can identify the smooth muscle. We find smooth muscle lining the digestive tract, lining the blood vessels. We don't have conscious control over it. It is an involuntary muscle. Cardiac muscle is found in the heart. Like skeletal muscle, it is striated, so you can see the little stripes. The best way to tell the difference is to look for these intercalated discs. These are regions where 2 muscle cells meet, and it allows them to send a signal from one to another and helps to hold them firmly together. Nervous tissue contains 2 major types of cells. Neural glial cells come in several different types, and they don't actually send signals. Instead, they regulate the environment, they provide a structural framework that helps to support the neurons. Neurons send signals. They typically have many branches that pick up signals. These are called dendrites, around a cell body with the nucleus, and then a long extension that transmits a signal. This axon may be very long, so when we look at a neuron, we see the projections, but we don't see the entire axon. Here's a neuron surrounded by some neural glia. Here's a smear showing several different neurons. And you cannot see the entire axon because they often transmit signals quite some distance away from the cell body. The remaining tissues are connective tissue. And even though blood and bone don't seem to have a lot in common, they do have some characteristics that allow them to all be classified as connective tissue. For one, they all arise from the same tissue in an embryo. They all arise from a tissue called mesenchyme. Within the tissue, we have specialized cells, but most of the volume is an extracellular matrix. In epithelium, it was cell next to cell. In muscle, it was cell next to cell. Here the cells are scattered in an extracellular matrix consisting of long proteins called fibers. These are not the same as muscle fibers, which are an entire cell. And then a ground substance. And that ground substance filling the space is different depending on the connective tissue. Connective tissue proper has a syrupy ground substance. The ground substance in fluid connective tissues like blood is more watery, while the supporting connective tissues have the firmest, most rigid type of ground substance. Connective tissue proper, with its syrupy ground substance, comes in 2 major types. Loose connective tissue has far fewer fibers than dense connective tissue. One type of loose connective tissue proper is areolar connective tissue. These are the scattered cells. These are the protein fibers, and this would be the syrupy ground substance. Areolar connective tissue looks like a bunch of little cobwebs or something because you've got all sorts of different sizes of fibers running in all different directions. Adipose tissue, the fat tissue that stores energy, is full of cells because the cells are enlarged. They become so full of stored fat that the cytoplasm and the nucleus end up squeezed to one side. So you end up with a bunch of empty-looking cells with fat inside of them. Dense regular connective tissue is filled with protein fibers. It looks kind of like skeletal muscle, but it's not striated. And these are not nuclei, these are the entire cells, squeezed to one side in between the fibers. Dense irregular connective tissue looks like a big jumbled mess because the fibers are going in all sorts of different directions. It's hard to even describe what that looks like. Fluid connective tissues have dissolved fibers so you can't see them in a watery ground substance. In blood, we mostly have red blood cells. In a mammal, a red blood cell does not have a nucleus, and a few white blood cells and small platelets. Lymph has more of the white blood cells. Bone is one of the supporting connective tissues. Its matrix is strengthened by calcium phosphate and other minerals. The basic structure of bone is an osteon, a little bullseye-looking thing made by layers or lamellae of bone matrix surrounding a central canal that contains blood vessels. In cartilage, the ground substance is a firm gel-like substance. Cartilage can be recognized by the cells, which are in little openings called lacunae. But these look like little eyeballs floating around inside. If you see those little eyeballs, you're looking at some type of cartilage. One of our cartilage types, hyaline cartilage, has fibers that don't stain very well. So when you look at it, it just looks like eyeballs floating in jello without any fibers. Elastic cartilage still has those cartilage eyes floating in their little openings, and it has wispy fibers that branch in all directions. The final type of cartilage is fibrocartilage. We still have the cells in their openings, but most of the extracellular material is filled with long wavy fibers. So fibrocartilage or fibrous cartilage is packed full of fibers. Those are the major tissue types, and hopefully you now have a better idea of how to identify them.